Well, good evening and welcome to this episode, a special episode of Dazzle Unlimited. I'm Steve Steinberg, the series host. We're recording this particular show in Stockholm, Sweden, early December 2014, and in conjunction with the world-renowned Nobel Prizes. And my special guest with me here, who's graciously accepted to do this interview despite our time pressures, is Nobel Prize winner W.E. Moerner. And you shared the Nobel Prize in chemistry with Dr. Betzig and Dr. Hell.
That's correct.
And I attended your lecture this morning. It was one of the Nobel lectures with thousands of people in attendance. And tremendous applause, by the way. It was fast-paced and fascinating. And you talked about not just general chemistry, but what is your word for it? Super
resolution.
Super resolution.
Right. Yes, the whole prize is about super resolution.
Explain that for our viewers who are not familiar with this.
I'm happy to do so. It turns out that there is a limitation on light microscopy that's been around since the late 1800s, called the diffraction limit. What this means is the tiniest object that you can see, because of this physical effect, is just half the wavelength of light. In other words, with visible light, that means about 200 or 250 nanometers. Now, of course, that's a very, very small number already.
That's about smaller than the width of a strand of human hair.
1,000 times smaller. Your human hair is about 100 microns, and this is a factor of 1,000 below that. But that diffraction limit is still large compared to what's inside a cell. The individual molecules, enzymes, and proteins, and so forth that we would like to watch are much smaller than this optical diffraction limit. So this has been a limit for a long time, a fundamental physical limit. So super resolution refers to methods to go beyond that diffraction limit and for the first time to see structures that are much smaller than 250 nanometers, down more to the scale of the size of the objects inside the cell.
Well, electron microscopes, for example, have been around for a long time. How do they fit into this scale of resolution, super resolution?
Yeah, that's a great question. Electron microscopes have, of course, better resolution, down to the few nanometer scale, but they require the cells to be killed. They do not allow you to look at live systems. And moreover, the image that comes from an electron microscope is just black and white. You don't know which object is there or not. You don't know what protein you're looking at. You see all of them at the same time.
Well, why is color so important?
It's not so much color, but you want to highlight the protein of interest because let's say a particular protein that makes a filament inside the cell, you'd like to have just those filament proteins be visible so you can see the shape of the filament. And that's what you can do with light. You can directly label just the molecules of the of the filament, so that you can see the filament itself. And now we can see it with this much better resolution rather than on a very fuzzy level.
But are we talking purely about optical resolution, or is there another category that you're working on or that this relates to?
We're talking about just the resolution by which you can see these objects, okay? And if you use only light, previously you were limited to 250 nanometers, so you couldn't see the fine structures. And think of it like looking down at a city from a very great height. You don't see much of what's going on in the city. But if you can be much closer, you can see the individual characters moving around and that sort of thing. And that's what this allows us to do. But with light, that doesn't damage the cell, and using the fact that we know which objects are being illuminated, because we use the fluorescence from the labeled object, or the light emitted from the labeled object.
And by labeled object, they're treated in a certain way so that they will fluoresce under certain conditions.
Yes, that's right. That's right. For example, we can use the technique of using the green fluorescent protein and its variants. This is a special protein that received a Nobel Prize some years ago that you can tell the cell to make the labels already attached to the proteins of interest that you want to image. And so that's one way to label. Other ways to label involve bringing in molecules from the outside that are fluorescent.
Can you actually see components of individual atoms with an optical system of this super resolution, or do you have to go to another system?
Well, the atomic scale is even smaller, down to, let's say, 1/10 of a nanometer. Remember that the limit before was 250 nanometers. And once you go below 250, you already see exciting things at 100 nanometers, at 50 nanometers, at 40 nanometers, 30 nanometers. We already see many factors below the diffraction limit. But an atom is way down there and much, much, much further. And ultimately, though, these methods should allow you to see at the level of individual molecules, which are about 1 or 2 nanometers in size.
I'm going to ask a dumb question, but you tell me if it's dumb, really. How important is this?
Well, it's not a dumb question at all. Of course, we, as scientists, want to learn everything about how the world works. But it turns out when you do that, when you can have the basic science approach, you also can learn things that can help, for example, affect disease, or help us learn how to deal with other problems that we might want to address. So, for example, one of the structures that we've been imaging in cells relates to Huntington's disease, which is one of the brain-wasting diseases that comes from aggregations of Huntington proteins. And with these methods, we can now see the shapes of these tiny fibrils inside cells at different times as the cell is progressing. And that's a way to learn something about parts of cell biology that were not accessible before. And so you can imagine that these methods will have impact in areas, not just that one, but any other area where you want to understand in detail how cells actually work, and also how they fail, how they misbehave. So these methods let you look and see where you couldn't see before.
Can you also see animated systems in process with this system?
Yes. So let me just emphasize that one of my contributions to all of this was to see single molecules, to be able to detect and image individual molecules. These individual, let's suppose you have one player inside the cell that's the only thing that's fluorescing, the only thing that can emit. You can see it moving around, you can see it binding to partners, you can see it doing things that it's going about its normal business.
Which is something that was not able to be seen before.
Correct, that's right, before we could see single molecules. The other laureates, Betzig and Hell and so forth, have developed methods to combine single molecules with the ways that you can image extended structures at very high spatial resolution. So it's really a revolution in optical microscopy that lets us see what we couldn't see.
It's interesting, I think you and your colleagues also commented on this to some extent in the lectures. This isn't per se purely chemistry, is it? Doesn't it get into physics and some other areas as well?
Well, absolutely. It turns out this is a very interdisciplinary prize. But let me just emphasize that there's an area of chemistry called physical chemistry, where you use physics to study molecules. And there's another area, synthetic chemistry, where you make molecules. And so chemistry includes a bunch of areas. But it's true that much of the scientific content in this prize relates to optics and how to generate these labels with chemistry, or optics in terms of how you use the microscope to look at the structures. So Professor Hell, for example, invented a special kind of microscope that goes below the diffraction limit, using some optical methods that involve switching the molecules on and off.
Steve Steinberg here with a program note. You are watching the television series DAZZLE Unlimited. You know, the series airs every Wednesday evening at 8:30 p.m. here on Time Warner Cable of New York City, Southern Manhattan, Channel 79 and 1081. Please make a note of it. By the way, if you have comments for us, or if you'd like to advertise on the DAZZLE Unlimited television series, contact us. The email address is dazzle@dazzleunlimited.com. Thanks. And now, please stay tuned because there's more to come on tonight's episode of DAZZLE Unlimited.
Start Heart Walk is coming to your area. Bring your family, bring your friends. Start something in your community and help save the lives of others. Join the movement and help fund research to fight heart disease and stroke. Start walking. Start something. Call 1-800-AHA-USA1 to find a Start Heart Walk near you or log on to heart.org/heartwalk to register.
I'm Ed Begley Jr., and I remember what my lungs felt like as a kid growing up and playing outside in polluted air. I'm concerned about air pollution, and I'm fighting for our right to breathe healthy air. That's why I volunteer with the American Lung Association. Too many people breathe too much dirty air on too many days across the country, which sends children and adults to the hospital at alarming rates. Together, we have the power to change that. Imagine how bad our air would be without the American Lung Association. Please support the American Lung Association and its fight for cleaner air. Take steps now to make sure future generations can breathe healthy air. Healthy air is vital to everyone. We all need it to breathe, live, and thrive. And if you're concerned about air pollution, asthma, lung cancer, and emphysema, get involved. Contact your nearest American Lung Association at lungusa.org or call 800-586-4872.
You see, a key aspect of these measurements require control of the molecules. You want to be able to have them be on sometimes and off other times.
When you say on and off.
Yeah, what I mean here is, you were talking about fluorescence. Fluorescence is when you pump a molecule and it gives you light back. Emission, okay, it emits light. You want to think of something like the day glow socks that people buy, or highlighters that people use to highlight things on paper, and you see that it has sort of a glowing aspect. Those are fluorescent molecules. And those are the kind of molecules that we actually can use for these experiments. But if you have them all emitting at the same time, you have a big blob. So you have to have ways to turn them on and off. And that's why critical to getting super resolution is this business of having them be in one form where they emit light when pumped, and another form where they don't emit light when pumped. That is, they're dark. And those are the two states we have to switch between to get super resolution.
It strikes me as, in one aspect, similar to, what is it called? But with the Earth Resources satellites for determining from orbit ground truth. The manipulation of different color filters, or could be gamma ray or X-ray filters, to bring out one feature versus the other in order to see what you want to see. Well, okay, that's my observation anyway.
Well, in this case, we don't necessarily have to use different colors. It's really the act of the molecules being emissive, able to emit, or not emissive, unable to emit. And that's a key way in which we're able to go beyond the diffraction limit. Remember that just one molecule is about a nanometer in size, but because of this diffraction limit, the light from it looks like it's about 250 nanometers in size. That's the problem of the diffraction. But if you have many molecules that are close together, they just overlap and blur. So we use this on-off mechanism to make sure only one is on at a time, and then we localize each one separately, and then put them all together in a pointillist reconstruction, very much like pointillist art, to show you what the underlying structure is. The points are where the single molecules are located.
I understand. My next question may be a little bit difficult to answer in layman's terms, but so that everybody can grasp this. What techniques, or can you give a general sense of what techniques and directions you had to go through to achieve this result of super resolution, super optical resolution?
Well, for my part of the story, which was the original detection of single molecules, how did I get to that in the first place is probably something that I can explain. I happened to be working at IBM Research, and at IBM we were interested in optical storage, using molecules, but not individual molecules. And it turns out we were going to record information by making changes in the emission of molecules, and whether molecules are present or not. And you could use that to record bits, 0 and 1 and so forth. And it was part of just exploring the fundamentals of that idea, the fundamental limits of that idea, that I realized we could detect a single molecule and then proceeded to detect it. So coming from the 80s, the late 80s, when the era of the great industrial research centers, there was a great balance between technology and fundamentals underlying the technology. And we discovered these single molecules there in that very different regime at low temperatures even. Later, this became room temperature, and so the idea of seeing a single, first at low, then at room temperature, controlling the molecules, was all put together with the ideas of the laureates to turn it into super resolution. You see, the original seeds were completely different in another area of science. We're simply trying to understand nature and understand fundamental limits. So that's an interesting aspect of how this arose.
And maybe the core of the beginning of this process and the pursuit of the process was that you, as well as the other winners that you shared this prize with, had, is it correct to say, a really driving curiosity to find where you could, how far you could go?
Well, absolutely. That of course, does drive a lot of us. We want to see things that are new, that haven't been seen before. We want to explore new regimes. When you turn a leaf and start seeing in a regime that you couldn't see before, surprises generally appear, and they did. We saw these molecules blinking on and off.
Was that something that was expected at the time?
No. That's what I mean. That's what I mean by it being such a surprise and a wonderful thing that shocks us. And then later we think of ways to use it. Okay, but part of the initial driving force is just to explore unexplored regimes where there's a good chance of finding these surprises.
One of your colleagues this morning in the presentation closed his lecture with exhorting members of the audience to have the drive, but to be okay with failing, maybe repeatedly failing.
That's right.
In order to, well, how would you view this?
Oh, I absolutely agree. It turns out when you're trying to do science at the frontier, you have to be ready to fail. We're doing something that hasn't been done before, so you don't know whether it's going to happen or not. You have to be prepared to have there be mistakes, and then when you learn from those mistakes, you move to another regime and keep moving forward. But you have to have an environment where you can explore things and not be afraid of failing over some extended time period. And this is pretty important for our science in general all over the world. We have to have continued stable funding for science and fundamental science to be able to explore new frontiers.
Is there a problem with that generally?
There's been some difficulties with funding. That is, there's been restrictions on funding and reductions in funding, smaller and smaller probabilities of getting funded per proposal that you write, down into the 10% and 15% range.
Is that
and
because
this makes it hard?
Is that because of the general world economic situation or something else?
Well, it's because of decisions as to what's important and where money gets allocated. That's really where it's coming from. And we have to make, well, we always make tough decisions in our society about where to spend money. But it is difficult. I'm just pointing out that it's difficult for these kind of fundamental science studies to have variable funding. It'd be much better to have more stable funding.
Before we started videotaping, I mentioned something and your response was, well, that's politics. Because of my suggestion, I don't remember what it was. But are you primarily, in this point about funding, talking about political situation?
Well, our elected leaders, of course, have a tough job to decide how to spend money, how large taxes should be, and so forth. But my only point is that I think there should be a continued stable place for fundamental scientific research, so that we have the opportunity and the freedom to come upon these kinds of discoveries.
Steve Steinberg here with a program note. You are watching the television series DAZZLE Unlimited. You know, the series airs every Wednesday evening at 8:30 p.m. here on Time Warner Cable of New York City, Southern Manhattan, Channel 79 and 1081. Please make a note of it. By the way, if you have comments for us, or if you'd like to advertise on the Dazzle Unlimited television series, contact us. The email address is dazzle@dazzleunlimited.com. Thanks. And now, please stay tuned because there's more to come on tonight's episode of Dazzle Unlimited.
These hands hold tight. These hands love. These hands support. These hands create. These hands save lives. To empower your hands to help make a difference, call the American Heart Association at 877-242-4277 for a CPR class near you. These hands are wise.
Explore new worlds. Read. Visit literacy.gov and let the journey begin.
Inspiration. Early on, or maybe throughout your experience in these areas, what has inspired you? What inspired you when you were a kid to say, this is my path. There are discoveries to be made. I'm going to go after them.
Yes, well, you know, it would be exciting to sort of say that, but I think what happened for me was a little bit different. I grew up in the 50s and 60s, at a time when Sputnik had shocked our world into young people needed to study science. And I was encouraged by my parents to study science. My father was trained in physics and chemistry, and my mother was a school teacher, and they supported everything that I did. So whether it was taking apart an old TV set, or building an amateur radio, or doing a chemistry experiment out of the shack in the backyard, okay, all of these things were supported and encouraged. And so that of course allowed me to play, if you like, and I really enjoyed it. I enjoyed trying to figure out how things work, or from another point of view, how to fix things. You have to have an understanding of how something works in order to fix it. And my father was a person who could fix almost anything, and I learned a lot of that from him. So part of my inspiration was from my parents, but at the same time there were great scientists, Einstein, Faraday, and so forth, who had influenced many fields. And I turned out to be more of this generalist who studied many fields in college, 3 bachelor's degrees, and have learned chemistry, and I've learned biology, and so forth. And so we're having a wonderful time exploring boundaries and trying to understand how the world works from all these points of view, because each of these techniques has a different special power in terms of trying to understand what's happening.
If you could, I think you probably have just covered what I'm about to ask anyway. So maybe there's a time reversal here. For our viewers who are listening to what you're saying, and they're saying, yeah, I would like to try something. I'm creative. What would you advise them?
Well, the advice I would give to people is to sort of maintain this passion of wanting to know how things work. Don't just accept that this phone is here and it'll do anything you want. You actually have to ask yourself, how does it really work on the inside? You can take examples from any of the scientific discoveries. For example, our single molecules or the LEDs that were awarded this year in physics, and ask yourself how they work. But at the same time, that passion is critical because it helps people get beyond the failures or the risks that lead to dead ends to be able to keep moving forward. So my advice is to learn many fields, actually, in order to be able to look at the boundaries and find the new exciting work that's at the boundaries between fields.
And maybe find the unexpected crossovers. And
Yes.
one thing generates interest and knowledge about something else, for example.
That's right. The synergy really that comes from using maybe a point of view from one field to try to solve a problem in a different field. But for the young people, you have to study science, you have to do your math, you have to do your homework, and learn a field deeply, and then add to that. I'm just so excited to be a lifetime student, to keep learning and learning more and more.
I think it's tremendous.
Millions of people are affected by disaster in America. And we're right there, providing food. With more than 200 food banks, reaching communities all across America. Donate now for immediate relief and long-term recovery. Every dollar helps provide food for disaster victims. Help now at feedingamerica.org.
Before a disaster turns your family's world upside down, it's up to you to be ready. Get a kit, make a plan, be informed today.
Anyway, thank you so much. My guest, William Moerner, W.E. Moerner, one of the 3 winners of this year's 2014 Nobel Prize in Chemistry. You shared it with, as I said, Dr. Hell and Dr. Betzig, and absolutely fascinating discussion. So thank you for being a guest. And thank you for joining us for this Dazzle Unlimited. I'll be back next week with another special guest on another Dazzle Unlimited, same time, same station. See you then. Good night.
This is Steve Steinberg speaking. If you have comments, suggestions, or would like to advertise on the Dazzle Unlimited television series, let us know. Our email address, dazzle@dazzleunlimited.com. The Dazzle Unlimited series airs every Wednesday evening at 8:30 p.m. here on Channel 79 of this system. Please make a note of it. Thanks for watching. Hope to see you again next time on another episode of Dazzle Unlimited.